This is Dr. Shanawaz again with a new topic on biotechnology or recombinant DNA technology. In this topic, I will try to explain what is biotechnology, and this topic is important for the NEET students, MBBS students, PG students. Before going ahead, I'm requesting you to subscribe this channel and share this video with your friends because sharing knowledge is an important thing and you will get reward for it. So, first of all, we will start and try to understand the outlines of this lecture. So, first, in this topic, we will explain the basic principle of recombinant DNA technology. What are the vectors and various types of vectors? What are blotting techniques? What is sequencing? PCR, its variant. And today's class will be 1 hour class. And after completion of this class, we will start. Then we will uh, make another video of 1 hour class, as this is a 3 hour class of biotechnology, and here you will learn a lot about this topic. So first of all, let us start with the basic principle of recombinant DNA technology. So what is the basic principle of recombinant DNA technology? So first of all, we will try to understand what is biotechnology. So the definition of biotechnology is that biotechnology is a technology that utilizes biological system, living living organisms are a part to develop or create different products. So this is a very important definition of biotechnology. That in biotechnology, what we will learn about, this is a technology in which we utilize biological system, that is living organisms are a part of biological system. What do you understand by the part of this? That means we can utilize different uh, parts, for example cells, tissues, to develop a new product. For example, what can be a new product? The new product can be this new product can be, it can be uh, vaccines. It can be vaccine, like you recently saw the COVID vaccine being designed using the biotechnological techniques. And so the new products can be antibiotics. So basically this biotechnology is the alliance between the biology and technology, and there is a alliance. It means the this part, bio part and technology part, has come together, and it has made a new subject, that is biotechnology. So it is an alliance. So modern biotechnology includes genetic manipulation, it includes cell fusion techniques and improvements made in old biotechnology processes. What are the old biotechnology processes? Like making of curd, making of cheese, these are the old biotechnology processes. Now with the new technology being utilized in this, and where organisms are, their parts are being utilized. For example, a new biotechnology is a CRISPR-Cas technology, where we perform the deletion of any unwanted gene so that the disease can be completely removed from the cell if there is a mutation in that gene, or we can introduce a new gene there. So these are the new technologies, and that's why there is an improvement made in the old biotechnology processes. So we will first then understand what is gene manipulation and cell fusion techniques in the later in this chapter. So basically this biotechnology is a technology that utilizes biological system and living organisms are part of the living organisms to develop or create new products. So this is a simple definition of biotechnology. First of all, then we will see the brief history of recombinant DNA technology. That means where from this has come and how this concept has developed. The two scientists were Herbert Boyer and Stanley Cohens. So what they did, Herbert Boyer and Stanley Cohens, what they did, they first developed the recombinant plasmid, and for example, they took the amphibian DNA duplex and they cut the rRNA a gene from it, and then introduced that gene into the plasmid 101, and then it combined it and make with it combines with the plasmid 101, the portion of this RNA combined with this plasmid 101, and it is now recombinant product. So this is the recombinant product. Who have first developed this recombinant DNA technology? It is Herbert Boyer and Stanley Cohen. Now, what is the basic principle of recombinant DNA technology? First of all, we need the DNA for it because it is a DNA technology. So we need the DNA for it. So isolation of DNA is important. First step that involves the extraction of DNA from organism that contains the gene of interest. So what is this gene of interest? For example, we need we know that some bacteria are producing proteins or antibiotics or any enzyme. That enzyme is very important for us, and this enzyme we need in bulk, and it has a industrial use. It has industrial usage. So the gene of this enzyme becomes gene of interest. So this becomes the gene of interest. So we will need this bacteria, we will isolate DNA, and then we will get the gene of interest from it. So this is the very important point. You have to remember it. What is a gene of interest? A gene of interest can be anything on which we want to do further biotechnological processes, or we want to do perform some biotechnological work. So then what we need? We got the DNA. Okay, we have the DNA here. Now we need the specific site. For example, this on this strand of the DNA we have the gene of interest here. This is the gene of interest here, and we need to cut it out. So how can we cut it out? What is the way to cut it out? We can use the scissor. And what are the scissors in biotechnology? We will discuss that later on. So we need, and where this scissor will cut, it will cut on the specific site. So what are the scissors? Scissors are restriction enzymes. So and restriction enzymes, they are called as restriction endonucleases. So these restriction endonucleases will cut this DNA. They will cut this DNA here, and we will get the strand of DNA that is cut. But they will cut at the specific site, and this specific site where the cut is made is called a restriction site. So you understand what is the restriction site and what is the specific site. This enzyme can cut in the two way. One way is called blunt end. This is the blunt end, and one way it can make a overhangs. When the enzyme cut in the form of overhangs, this is called a sticky end. So one is blunt end, another is sticky end. Enzyme can cut the DNA up and down, that is called as a blunt end. Enzyme can cut DNA at one point, and another strand can be cut at the another site. That's called, then it will make the overhangs. These overhangs are, this DNA that is cut with the overhangs is called as a sticky site. It is called as a sticky site. So the enzymes can cut in two way. One, it can produce the blunt ended DNA, or it can produce a sticky end DNA. Now, after that, we have now what we have got. We have got the DNA. We have got the two. We have got the gene of interest from that DNA. First of all, we took the cells. We got the bacterial cells, and we isolated the DNA from it. And that DNA, by using the restriction endonucleases, we made the cut at the specific site, and we got the gene of interest. This is the gene of interest. This is the gene of interest. Now we got this gene of interest by using the restriction endonucleases. Now we have the another DNA that is called as a vector. So DNA fragment generated by restriction enzymes are inserted into the vector. This vector is naturally, this vector is plasmid in bacteria. In bacteria, and restriction enzymes can cut this vector, and if this vector is cut, we can insert this gene of interest here, and this is called now recombinant DNA, or the second name is chimeric DNA. Now, what is this vector? Vector is a DNA molecule. It is also a DNA molecule. This vector is also a DNA molecule. It is also a DNA molecule. This is also a DNA molecule that carry foreign DNA. That carries foreign DNA. Where it carries this foreign DNA? Now, after introducing this plasmid or gene of interest into the vector, that is plasmid, we can now introduce it into the competent cell, and it can be DH alpha can be the competent cell. This vector will go inside this competent cell, and this cell will then clone. This cell will then clone it, and we will get the uh, multiple copies of this gene of interest. So, common vectors are circular molecules and bacteriophage, that is the virus that infects the bacteria. These are the common vectors. Now, we are in this uh, first, when I told you that we need the Caesars. So Caesars are the restriction endonucleases. We don't need really Caesars to cut this DNA, but we have the restriction endonucleases that can be used to cut this DNA. Now we need the glue as well. So what is the glue in case of recombinant DNA technology? And this glue is DNA ligase. DNA ligase acts as the glue. It acts as the glue. That means when we have the DNA, plasmid DNA, and we want to introduce this gene of interest into this plasmid DNA, for joining this DNA with the plasmid DNA, gene of interest with the plasmid DNA, we need the enzyme that is DNA ligase. DNA ligase. We need the enzyme DNA ligase. So this DNA ligase, it seals the nick in sugar phosphate backbone. In sugar phosphate backbone. So this is the basic principle of DNA recombinant technology, RDT. That what are the steps in it? First we need to isolate the DNA. The isolation of DNA is followed by the cutting the specific site of the DNA, then inserting the DNA fragment into the vector and joining the fragments. So this is the donor cell, which gives us the gene of interest. These are the foreign DNA, and by using the restriction endonucleases, we will cut these DNA, and this is the plasmid from the bacteria, and we will use the ligase enzyme to join this foreign DNA with this plasmid or a vector, and then formation of the recombinant DNA is followed by the introduction into the host bacterium with the recombinant DNA. That means this host bacteria, this D vector will go inside the host bacteria, and then we will also discuss how it goes inside the host bacteria. That is called as a competent cell, which takes the host bacteria, competent cell, and then it will form the multiple clones of RNA, recombinant DNA, and clone is engineered bacteria in the medium, and then recombinant product we get if these recombinant clones will be used to get the product, and then we will perform the downstreaming and separation and purification of the product. So then it is the introduction of the recombinant DNA into the host cell, and then this process is called as the transformation, and the host cell takes up the recombinant DNA and starts to replicate it. Then there is selecting the transformed cell. Not all host cells will take up the recombinant DNA. Some will take, for example, we have multiple host cells here. Some host cells, some host cells will take this vector, and some will not take this vector. For example, these three took this vector inside, and some didn't took this vector inside. For that, we use the selective markers. Selective markers such as antibiotic resistance genes, and that can be used to identify and select a cell that has successfully integrated the recombinant DNA. Now, then after that, there is one more thing, that is cloning and expression of the DNA. One is the recombinant DNA is inside the host cell. These cells can be cultured to produce many copies or clones, and if the goal is to produce a protein, the host cell will express the gene carried by recombinant DNA and produce the desired protein. We will get the desired protein. Then there is screening of and analysis. Finally, recombinant DNA or the expressive protein is screened whether it is the correct that protein which we need. Analyze it to confirm the correct gene has been inserted and function is expected. And the techniques such as polymerase, chain reaction, gel electrophoresis, and sequencing is used for this purpose. Now, we will try to understand what is restriction endonucleases. In the principle of basic principle of recombinant DNA technology, whatever we have discussed, we will now try to understand that bit by bit so that there will be no confusion in it. Now, first of all, we will try to understand what is restriction endonucleases. So, restriction endonucleases are the bacterial enzymes. They are the bacterial enzymes that cleave the DNA selectively at a palindromic sequence of 4 or 8 nucleotides. This should be understood. What is this palindromic sequence and how many nucleotides are this sequence is made of? For example, if you will check this word, this is Malayalam. If we will start reading it from this side, we can read it correctly from this side. If we will try to read this DNA from this side, it is AND. Now if we will read this Malayalam from this side, we can easily understand that it can be read from this side, it can be read from the backward and forward side. That is Malayam. That is called as a palindromic sequence, and this palindromic sequence can be of 4 or 8 nucleotides. This is the first discovered in E. coli restriction replication of the bacteriophage by cutting the viral DNA. First, when we come to understand about the restriction in the nucleus, they were first discovered in E. coli. What does this E. coli did? The bacteriophage are the viruses that eat up the E. coli. So E. coli has these are the enemies. Bacteriophage are the enemies of the bacteria, and this is the E. coli. And this E. coli has developed a mechanism. It has a mechanism that if bacteriophage introduces his DNA, its DNA into this E. coli has the restriction endonucleases, and they cut down the bacteriophage viral DNA and saves the life of its own cell. That means this now bacteria will live a long life. It will not die. So they were first discovered in E. coli, restricting the replication of bacteriophage. So this bacteriophage dies inside the E. coli. But the bacteriophage are the viruses that eat the bacteria. That's why they are called as the bacteriophage. Now, by cutting the viral DNA, thus the enzyme that restricts the viral replication is known as restriction enzyme. That's why we got this name, restriction enzyme. If you have confusion why we are calling it as the restriction enzyme, it's called as the restriction enzyme because these enzymes restrict the viral replication, or restriction endonucleases. Endonucleases we will because they cut the DNA in between the DNA. They didn't cut the DNA from the end side of the DNA, that is outer side of the DNA, that is exonuclease. It was discovered by Werner Arber. So the first letter. Now we will try to understand what is the name, how we name these restriction endonucleases, and what is the way to name them, the restriction endonuclease. So first of all, what is the restriction endonuclease that is released by E. coli? There are multiple restriction endonuclease released by E. coli. We will try to understand ECO R1. So, what is this ECO R1? The first letter E stands for the genus name of organism or bacterium. So, it is always that you should remember it to capital, write in a capital form. It should be capitalized. That means we will write the capital E and isolated, and this is the which is the enzyme isolated and capitalizer. So that is capitalized. E stands for E. coli. Now in eco R1 it is E. coli, that is a genus name. Like another restriction enzyme, that is hin D third, that is H stands for Haemophilus genus. That means this restriction endonuclease is isolated from Haemophilus, and this is the genus name of the organism. In the same way, BAM H1 is Bacillus. That is B stands for BAM H1. Now there is another word, that is IN co and AM. What is this second name? This the next two letters are the two letters of the first species name. That means E. coli. In case of E. coli, Escherichia coli, this co is from coli. This co is from coli, and like co is the coli species. Hin is the influenza species. That is, we used the first two letters of the species name. And am in case of BAM H1 is amylo liquefacient species. That means amylo liquefacient species, we got this am from it. It should be remembered, and it is mostly asked in the exams. Now the third is R. What is this R? R is the strain, that is strain designation. It is applicable that the next letter may represent the strain of the bacterium, and R is the strain, and there is no strain designation. But in Hind third, D is refers to the strain R. D. Haemophilus influenzae. That is Haemophilus influenzae, which gives the hemophilia. That is RD strain, and that is what we call it R. R means no strain, and in case of D stands for RD strain, and in case of H stands for H strain. That means strain. Now there is a Roman letter, that is one third, one BAM H1, that what is this Roman letter. So Roman numbers are used to indicate the order in which the enzyme was discovered in order. That is this is the first enzyme that was isolated. It can be now the second enzyme which was isolated can be R2. Third enzyme can be eco R3. And in this word, it is the order. It tells that this hin D third is the third enzyme isolated from the Haemophilus influenzae, and is the first enzyme that is isolated from this strain. So this is the first enzyme discovered from this organism, third enzyme discovered from this organism, and the first enzyme discovered from this organism. Now, there are three types, in fact, four types of the restriction endonucleases. One type 1 is the one enzyme with different subunits for recognition and cleaves. It causes the cleavage and methylation. It is very important. This sometimes asked in the exam, and this recognizes and methylates a single sequence but cleaves the DNA up to 1,000 base pairs away. That means that it cleaves the DNA up to 1,000 base pairs away from the restriction site. The second is the two different enzymes which recognize the same target sequence, which is symmetrical, and two enzymes either cleave or modify the recognition sequence. They are either cleave and they can also cause the modification of the recognition sequence. Now, third type one enzyme with different subunits, one recognizes and modification, and one for cleave. It is a two different subunits. There are subunits in these enzymes. This enzyme has two different enzymes with both which recognize the same target and which is symmetrical. And the third, this type second is a two bit recognize the same asymmetric sequence. They two different enzymes but recognizes say sequence is the recognition sequence is asymmetric. Type second recognizes symmetric, and second S recognizes asymmetric, and cleavage occurs other side of the on the one side of the recognition sequence up to 20 base pairs. So this is maintained by England Biolab, which tells about the types of the restriction endonuclease. So these are the restriction endonucleases, and this is the historical background that Arbor showed that certain enzymes of the bacteria restricts entry into the phage, hence they are called as restriction endonuclease. Hamilton Smith in 1970 isolated the first restriction enzyme that is called beta in D1, and Daniel Nathan in 1971 was the first applied the enzymes to cut the DNA. This is very important, and all three got the Nobel Prize. And Paul Berg developed the cutting technique of DNA recombinant technology. These are the persons: Paul Berg, Hamilton Smith, Werner Arbor, and Daniel Nathans. So you have to remember what was the role of the Hamilton Smith. He first isolated the enzyme beta in D1 in 1970, and Daniel Nathan was the first time applied the enzymes to cut the DNA, and all the three got the Nobel Prize in 1978. So, assuming that DNA molecule with 50% GC content and random distribution of four bases and four recognition sites occur in every 44 base pairs. So, what is the percentage of this base pair recognition, and what's the role of this GC content? That most of these and DNA molecules with 50% is the GC content, and a random distribution of four base pairs. And similarly, six base recognition sites occur every 46 base pairs. That means the four bases recognition site occur at every 44 base pairs. So that means in a DNA, how often these restriction recognition sites come. That is, if it's a four base pair, then it will come after 44 base pairs, and if it is the six base pair recognition site, it occurs every 46 base pairs, and eight base pair recognition sequence every 48 base pairs. In practice, there is not a random distribution of four base pair. Many organisms can be ATR GC rich, and the nuclear genome of the human is 40% GC, and the dinucleotide Cg is fivefold less common statistically expected. So these are the average sizes of these different enzymes with the different forms of DNA sources. For example, if the enzyme is APA1, that is in Arabidopsis, the length is 5. The average fragment size is produced by different enzymes is 25,000. It is 40,000, 6,000, 15,000, 2,000, and this is a different from the different sources. This same restriction enzyme produces different types of sizes. So this is the genomic DNA, and we use the restriction endonuclease to cut it down. Then we perform the gel electrophoresis. The long DNA fragments will be on the negative side, and the shortest will be on the positive side. Then denature in alkali and blot the transfer, that is in nitrocellulose membrane, and this we will get we will get only those who will pass this nitrocellulose membrane, and then hybridize with the radioactive probes, and we will wash it down. And then after getting transferred on the nitrocellulose membrane, we will perform the autoradiography, where we will find the DNA images of this gel. This is a recut with the PST, and this is a PST digest, and this is recut with the PST1, and this is the ligation side. This is how we recognize the DNA. Now in the next video, we will start understanding what is recognition site, and some restrictions and with the sources and recognition sites. And it is all for today. I told you that we will continue for the 1 hour, but we will start with the 20 minutes video now. I thought it will become the lengthy video, and it will be very difficult to watch it out. And in the next video, very soon we will start understanding